Cytotoxic and anti-Alzheimer evaluation of novel Lancifolamide isolated from methanol extract of Conocarpus lancifolius. In the Holy Quran, it is said, "Your God is that who has made different kinds of orchids and gardens for you. Those have colorful crops of seeds, Dactylifera, Olia europea, and Tunica granatum." Like in such references are found in Hadith, which emphasize on importance of plants and its use in human ailment treatment. There are many notable drugs which have plant origin, like aspirin from Salix alba and atropine from Atropa belladonna. Conocarpus lancifolius, the plant under in discussion. Here is the aerial view. Botanical classification of Conocarpus lancifolius is shared with you in this slide. The family of this plant is Combretaceae, genus Conocarpus, and species Lancifolius. Plant was selected from the peripheral areas of Lahore, and it was confirmed by a taxonomist in Bahadur University. Grinded and dried parts of dried and grinded plant materials of Conocarpus lancifolius were extracted with organic solvents such as dichloromethane and methanol through successive maceration and filtration. Oxidation was performed. Rotary evaporation was done to concentrate the extract. Aerial plants were collected, powdered, and dried. About 900 grams were used and both extracted with both solvents, dichloromethane and methanol. It gave us 12 grams of extract from dichloromethane and 19 grams of extract was collected with methanol. Preliminary phytochemical characterization was done and it gave positive results for saponins, steroids, flavonoids, cardiglycosides, and tannins. Here we have done LD50 value. We have taken LD50 value by doing brine shrimp lethality bioassay. Dichloromethane and the methanol extract were separately used, and the LD50 value of the dichloromethane extract was quite high, which was had less potency, whereas methanolic extract was quite potent. DPPH radical scavenging activity was done. Positive was used as the standard. The extract gave both extracts of dichloromethane and methanol gave significant results of DPPH activity as compared to the standard positive. Acetylcholine and esterase inhibitory was also quite significant when it was compared with the activity of standard, which was aspirin. Ex vivo screening of plant extract on isolated tissue preparation. The aqueous ethanol extract of Cornus lancifolius relaxed the spontaneous contraction in isolated rabbit jejunum preparation in dose-dependent manner. Potassium-induced contraction in isolated rabbit jejunum preparation indicates that the blockage was mediated by voltage-dependent calcium channel. It also relaxed carbachol-induced contraction in isolated rabbit tracheal preparation in dose-dependent. Here is the graph showing the result. Isolation. Cytotoxic assay-guided isolation was done by using column chromatography. Here is the protocol. Initially, the methanol extract of Conocarpus lancifolius of about 10 grams in weight was taken. It was done on TSE plate with a pore size of 40 to 63 micrometers. And the mobile phase selected had three solvents in it: dichloromethane, methanol, and water. Initially, 85, 15, and 1 ratio was used, and the concentration was changed as such that we moved towards 100% methanol. 4 sections were obtained. The second section, CLM2, was promising, which was extracted further, and in such a manner, we got a pure compound in CLM2 C5. 60 milligram of lancifolamide was extracted in a pure. Spectroscopy was done to check the structure of the isolated compound. Initially, UV spectroscopy was done, then IR, and the proton NMR was also performed. Physical parameters of compound extracted are: physically, it is colorless amorphous solid in appearance. Quantity was 60 milligram, which was extracted. Melting point 210 to 220 degrees centigrade. Ultraviolet radiation, the lambda max was 210 and 230 nanometers. Moreover, FTIR and proton NMR results are also given in this, are shared in this result for structural isolation. The structure device is such. The IUPAC name is also given here. Biological activities of isolated compounds. Crude extract was used. Cytotoxic assay was done by using the cell line. ED50 less than 20 micrograms per ml were considered active for extract and less than 4 micrograms per ml for pure compound. Murine lymphocyte leukemia, human colon cancer breast cell lines, and human breast cancer cell lines were also used. Molecular docking was done. Comparison of the binding modes of ellipticine and lancifolamide for selected proteins were done. Here are the results of cell docking, and various proteins which were docked are also shown here. Acetylcholine and acetylase inhibitory was also got for methanolic extract. Here are the results of the lancifolamide, DPPH and acetylcholine acetylase anti-acetylcholine acetylase activity. The first slide we showed, that was for extract, and now we are discussing particularly for the lancifolamide, the compound which we have extracted from our plant. The IC50 value of from the DPPH assay activity gave 184.32 the results, which was quite higher when compared with the standard process. So was the acetylcholine acetylase inhibitory activity was also significant, though it was lesser than the results of the standard use. To gauge the anti-Alzheimer activity, and we used a procedure which is called step-2 method. What is the step-2 method? Mice and rats of either sex are used. The test apparatus consists of a small chamber connected to a larger dark chamber via door. The test animals are given an acquisition trial, their acquisition trial. In the acquisition trial, the animal is placed in an illuminated compartment at a maximum distance from the door, and the latency to enter the dark compartment is measured. The threshold was of 90 seconds to 180 seconds. Animals that do not step through the door were rejected. Conclusion drawn from the research is, in this conclusion, the current study isolation of new compounds S4 isopropyl 8-methoxy 6-bicyclo benzothioate from methanol extract of C. lancifolius. Isolated compound showed prominent and anti-cancer, anti-Alzheimer activities, which give a clue about its immunomodulatory attributes. Molecular docking studies showed that lancifolamide induced cytotoxicity through the induction of apoptosis in the cancer. Further studies to standardize the methanolic extract and long-term toxicological studies are warranted to establish the therapeutic potential of this novel compound. Thank you very much for your attention.